Due to shortage of reagents in labs performing genome sequencing are facing problems. According to an official in the know of development, 5 labs have stopped genome sequencing due to deficiency of reagents. The officials also added that there is a decline of 40% samples being sequenced as compared to last month. In fact, let me go across to Ayushmann Kumar, who has got this exclusive information for us. Ayushmann, first of all, help us break it down for our viewers. What exactly are we talking about when we say that the labs are facing shortage of reagents? What role do they play in the process of genome sequencing, and who is supposed to supply them to the labs?
Well, currently what we have been told from the officials is that over 25,000 samples have been genomically sequenced. These are the COVID samples which were sequenced to see if the Omicron variant's number in India is rising. So the mutation, the COVID samples, positive samples are sent to the labs for genomic sequencing to assess as to what is the prevailing predominant variant that is present in the population, like we saw in Delta and in Omicron. Currently, the officials have told us that for genomic sequencing, a set of chemical is used which facilitates the genomic sequencing, that is called reagent. And that reagent is something that is not available to majority of the labs, and it is due to the shortage of fund. The shortage is to such an extent that 5 labs have stopped genomic sequencing. India has got 38 labs which are performing the genomic sequencing. 5 of them have stopped genomic sequencing right now, resulting in 40% drop of the number as compared to the previous month. Also, the NCDC, which is one of the nodal centers for genomic sequencing, is also facing the heat of the lack of reagent due to the lack of fund. But there the genomic sequencing is still being done. But, to assess the impact of a variant or the prevalence of a variant, genomic sequencing plays an important role. And here we are being told by the officials that because of the shortage of fund, the reagents are not being procured, and that is resulting in closure of the labs and also resulting in a decline in the number of genomic sequencing being daily done in India.
Right, Ayushmann, thank you so much for getting us all those details and also this exclusive news break. In fact, I am now being joined by Dr. Rajiv Das Gupta, Chairperson, Center of Social Medicine and Community Health, also JNU, and member of National AEFI Committee. Dr. Das Gupta, thank you so much for speaking to us and taking the time out. First of all, your reaction on this big news break on Mirror Now, where we are reporting that 5 labs out of 38 have stopped genome sequencing due to shortage of reagents. How do you see this particular development? How big a blow is this for India's COVID fight back?
Thank you. And I understand this is a ground report which is based on actual collection of what data on what's happening in these labs rather than any press handout. So if this is true, what we don't know whether it's a temporary glitch for a few days or it's actually deeper than that. Either way, genome sequencing is extremely crucial, particularly to understand the kind of shifts in the variants that's happening within the country. India had done well to expand this INSACOG network, the network of laboratories that contribute to the genome sequencing. We have seen how crucial genome sequencing is in the matter that South Africa was actually able to pick up the Omicron variant pretty early. So therefore, genome sequencing is a very key component, and to the extent that in several of his most recent addresses, the Prime Minister actually stressed on this. So that makes it doubly serious. However, it must be underscored that genome sequencing is a complex process. It requires high-end laboratory, high-end human resource skills, and all countries in the world are, relatively speaking, struggling to do adequate genome sequencing. But this glitch or this blip on account of fund shortages, as the report indicates, that is something more serious, of course.
Dr. Dasgupta, you mentioned the issue of funds shortage. In fact, our correspondent Ayushmann was also reporting the same thing, that because of shortage of funds, the reagents are not being procured by these labs, and hence the entire process of genome sequencing has stopped in 5 labs out of 38 that are functional right now in the country. But earlier in the past, we have also seen, Dr. Dasgupta, that several states on their own have stopped the process of genome sequencing, be it Delhi or Maharashtra, when it comes to Omicron samples specifically. And they had cited the issue of costly process. Do you think that in order to continue the process of genome sequencing without any hurdle, without any hiccups, it is very important to maintain the continuous flow of funds to these labs, to this entire system, and an able support of states and other governments are also needed?
No, first let me clarify the information of funds, information on shortage of funds came from the report filed by Ayushmann. I do not have any information on it, so I was just referring to his report. However, yes, genome sequencing is even per sample genome sequencing is an relatively expensive test than many other tests. There's a global benchmark of sequencing 5% of the samples, but that was in the earlier phases of the epidemic with such large numbers of Omicron led surge. That may not be that need not be a gold standard, in fact. Also, the genome sequencing is not relevant to individual treatment of patients, but for epidemiological tracking of what's happening. And in India, the large and diverse country that we are, it is understandable that different states or different regions will face these transitions from, say, a Delta to an Omicron in somewhat different time frames. And therefore, genome sequencing provides that data and that understanding. But at the same time, genome sequencing data interpretation is done over a time frame of a week, basically on a weekly basis. So these shifts actually become evident over a week or a fortnight or even a month. So if it is, to be fair, if it is the case that it's just stopped for a couple of days and the labs can make up whatever samples there are pending, that also should suffice over a week's in a week's time frame period, that is.
Right, Dr. Rajiv Dasgupta, thank you so much for taking the time out and speaking to us. In fact, let me go across to Neeraj Gupta now, who is a public health expert. Dr. Gupta, thank you so much for speaking to us. First of all, your reaction on this big news break where we are reporting that 5 labs out of 38 that perform genome sequencing have stopped the process because of shortage of reagents. And our correspondent was saying that it is because of shortage of funds. How do you read this particular development? How big a blow or how big a roadblock is this?
First of all, thank you very much. I would say that this is definitely, genome, first of all, we need to understand that genome sequencing is an important study which we do from the epidemiological study point of view. It doesn't affect the patient treatment or as Dr. Rajiv Dasgupta was also saying, that it doesn't affect the patient treatment or even the course of strategy which we have applied for the country in terms of how to deal with this pandemic wave. But at the same time, you have to also understand this genome sequencing is something which, when we know that by and large our country is experiencing an Omicron wave, it doesn't really make a difference if we do 50,000 samples or 25,000 samples. It is important to do genome sequencing, so we keep on studying and keep a tap on terms of if there is any new variants emerging, any new combination of variants that is emerging in our set of population. But if it's just to distinguish between how many Omicrons and how many standard other variants of the COVID in the previously known, it doesn't really make a big difference in our course of treatment or the course of strategy, how do we deal with this pandemic. Even if we have a little delays, as Dr. Rajiv said, that for a week, because genome sequencing technology can process large number of samples at the very same time. it is better to do larger set of samples together rather than lower number of samples in each run, because it reduces the cost, which is also a very important aspect of the process. So it doesn't make a big difference if it's delayed by a few days or a week or so. Definitely, if there is any shortage of funds as so far reported by you, as said by some authorities, then that should be definitely taken care of if possible, as per the needs. And it is important to do genome sequencing, but it is also very important to know that after we know that the major wave is already Omicron, it doesn't really help in bringing any additional value to the process. Most of the countries like in Europe, like in UK, has also did earlier more than 50% of their samples they were doing genome sequencing, but now they have brought down to a very large percentage, very small percentage I mean to say, from the large percentage. So because they know that if it's all Omicron, then we don't need to keep doing sequencing until less we know that there is something new coming up, for which we can do a smaller set of samples and keep doing that process.
Right, Mr. Gupta, when it comes to tackling COVID surge of any kind, not just in India but across the world, we have seen that the entire focus mostly remains on the containment measures, the restrictions, the compliances, the public awareness. While all of them are very, very important, of course, do you think amidst all this, the focus on research, on scientific assessment gets compromised in any way?
Probably not, I would say, because there are two separate segments which what all the governments which across the world works on is first how to make the immediate measures to restrict the spread and to bring appropriate medical treatments and strategies in place. But at the same time, the teams which are doing such sequencing project programs, so genome sequencing is the only tool which can help us determine if there is any new pandemic variant which is any new variant which is emerging out. And for that, we keep doing certain set of population. So the epidemiological teams which are already there existing, which work separately from all the other branches of the pandemic team, task forces, they keep on keeping a tap in terms of if there is anything new emerging in the market space, in their samples in the specific region or in the country. And if that keeps on happening, and we keep knowing that there is some new variant which is coming out, and then we keep a tap on terms of, because then the restrictions become the major criteria, because if there is any new variant that emerges in any other country, and if we bring appropriate restrictions to bring people coming in from that country, and if we can prevent them from that from happening, then it can help the delay or complete prevention from that wave coming into our country. So I think that's the first most step which any government should do, and they mostly like to do that. So just in case that happened in South African Omicron variant outbreak, people knew that it's coming from South Africa, so they all tried to bring in restrictions for passengers traveling in. They did, like in India, we did testing for all the people coming from flights from those regions, and we were able to delay or to minimize the wave as much as we can, as compared to many other parts of the Europe or US, for example, where they see a tsunami of positive samples for Omicron coming out.
Right, Neeraj Gupta, thank you so much for taking the time out and speaking to us on this big story that we are tracking on Mirror Now. Sources have told Mirror Now that the genome sequencing process in the country has hit a roadblock, and in fact 5 labs out of 38 which perform the process of genome sequencing have stopped functioning, and this is because of shortage of reagents. We will be tracking all the developments taking place on this big story.